Welcome back, friends. In this video tutorial we'll be talking about the Streptococcus species, and we'll be talking about several types of Streptococcus. In this first video we'll be talking about the introductory part about the Streptococcus bacteria. Now Streptococcus, as the term suggests, coccus, it means they are consisting of the spherical shaped bacteria, right? So they are spherical bacteria. So let me write, they are spherical in nature, right? So they are spherical in nature, and obviously they are gram positive. So they are gram positive in nature. Okay, so they are gram positive spherical shaped bacteria. That means they are cocci, right? So we can write that they are cocci or cocci, whatever. Now in this case of coccus, what we know that the streptococcus, it should look like something like that, right? So it's a spherical shaped bacteria. Now in under the microscope we never find bacteria separately placed like that. Instead they are attached with each other to make a chain like structure like that, right? It's a chain, it's a bacterial chain like that. Okay, now we can find the streptococcus often like that. Okay, and these are also free living bacteria. So let me write this property also, they are also free living, so no confusion about that, free living bacteria. Okay. Okay, now among the streptococcus there are certain species which are very much important. Now the one of the most important species among all is Streptococcus pyogenes. Streptococcus pyogenes, or Pyogenes, pyogenes. This is going to cause severe diseases in human beings. So that's why this is medically important. Except for that, there are Streptococcus bovis. So let me write the names. Streptococcus bovis, bovis. Then there are Streptococcus mutans, and obviously there are Streptococcus pneumoniae, which is a causative agent for pneumonia, right? Pneumoniae. So just focus on the spelling for the bacteria, and all of them are scientific names, so you should put underline in all these cases. Okay, I am not putting here for lack of time. Okay, now these are the different types of streptococcus. Among them, this one states stool, because streptococcus pyogenes is a very dangerous agent. Now the streptococcus pyogenes is a very dangerous because they can cause severe diseases, right? Now, the Streptococcus pyogenes usually cause diseases with the help of a protein which is called a M protein. So it is having something called M protein. Using this M protein, the Streptococcus pyogenes can cause infections in human being. Now, if I provide you the structure of the Streptococcus pyogenes, what is going to be the structure of Streptococcus pyogenes? Simply, it is telling us that something like that. So it is having a cell wall, and so let me draw the cell walls or cell membrane first. So if this is the cell membrane, for example, this is the cell membrane. Now let's draw the cell wall, and let's take this color. Okay, this one. This is the cell wall, for example, and outside the cell wall there is a capsule, and here we go, the capsule. And throughout all this place there are so let me draw. This is the M protein, for example. This M protein will have a structure like that. Say this is M protein coming out like this. Now obviously from other side another round of M protein is coming out. This is another round of M protein, right? So this blue colored protein here is the M protein of Streptococcus. Okay, and it is embedded through this cell wall system. And as it is embedding this particular terminal here, this terminal E is called C terminal of this protein, and this is N terminal of that protein. Okay, and it is embedded, right? And in this case, this particular protein, this M protein is the major virulent factor. This is the major virulent factor for the Streptococcus pyogenes bacteria. Okay, and what they can do, that they can trigger immune response, they can trigger different chemical toxins or cytotoxins, they can kill cells, right? And the unique feature about all the Streptococcus species, different types of Streptococcus species, that some of them are hemolytic, right? Some of them are hemolytic. Hemolytic. Now, what do you mean by hemolytic? As the term suggests, hem means definitely it is something to do with red blood cell, right? So hemolytic means it can, so it means it can uh, degrade degrade red blood cell, right? So that's what I mean by hemolytic. Now in this case, what we can see that if we plate this Streptococcus bacteria in blood agar, so let us draw this. So let us draw this. Let us take this one. So if this is the agar or petri plate, for example, now here we are having the blood agar coming out. So let's draw the blood agar in this case. Put it here. Okay. So here we are having the blood agar. Now here somewhere, if we start, so we inoculate this particular thing with our Streptococcus, after delivering some time, what it will generate colonies like that. So it will generate colonies like that. So here it is, colonies will be formed like that, which will be surrounded by the white zone, or sometimes colonies are also produced surrounded by this green colored zone in blood agar. So this kind of thing can be seen in this case. Now this white zone formation can be explained because this Streptococcus is hemolytic, so it lies down all the red blood cells that are surrounding that particular colony. So the colonies that give rise, so let me draw the colonies. I'm drawing the colonies with this color. So colonies are developed, and during this development of colonies, they are degrading certain position or certain number of red blood cells surrounding that colony, and it is creating this particular white ring surrounding that colony, right? So this is a typical characteristic. So it's a typical colony pattern, typical colony pattern for Streptococcus species that we can observe, right? But not all the Streptococcus are hemolytic. One of the example of the hemolytic Streptococcus is this Streptococcus pyogenes. Again, this is an example of hemolytic Streptococcus. Okay, and another thing is that this is one way of identifying Streptococcus. Obviously, there are other ways, like this is catalase positive, and also there is a test called Latex Kit Test. So let me write. This test is called Latex Kit Test. Now this Latex Kit Test, what we can do, that there are latex beads or latex, what you can say, small latex particles out there. Now if we put the small latex particle and along with the bacterial culture, now if it is a Streptococcus, in those cases what it will do, it will agglutinate those latex. So let me draw this structure for you. So if, let's say, these are the latex particles, so we provide the latex particles there. Now if, and what we provide, we also provide the bacterial culture. So let me write, culture is also provided here. Now after that, two conclusions can be, two different way or results can be obtained. One result is that all of these latex particle will agglutinate with one another, and on the other hand, another result can be there where they will be separated without being agglutinated. So if the second position occurs, in those cases what it provides us, a dark white, or it's a bright white, sorry, they provide a bright white solution, and in this case it won't provide anything. It shows, and this agglutination means its taste is positive, and unagglutination or non-agglutination means the taste is negative. Now positive taste means there is, that the bacterial culture we use is of Streptococcus, right? So if this thing occurs, we'll be telling that yes, the culture we'll use is eventually Streptococcus. If it never results, if it is not resulting in this kind of agglutination, then what we can say that yes, whatever culture we've used definitely is not Streptococcus. Okay, so that's how we can actually identify the different types of Streptococcus, and actually different depending upon their serological means. We can also divide them into several different sections, different serotypes actually, like alpha, beta, and all these things. I am not talking about them in this particular video, but these Streptococcus are important because they are clinically significant and they can cause several different types of infections, and we'll be seeing it in the later video. Thank you. So we have talked about the basic features of Streptococcus species bacteria. Now in this video we'll be talking about the clinical significance of Streptococcus species and different types of infections and diseases that are caused by Streptococcus species. Now I've told you that Streptococcus is also a serious concern like Staphylococcus. Now there are several different types of diseases and different clinical conditions are caused due to the Streptococcus infection, but here I'll be talking about only major four different diseases. So let us talk about that. So if we talk, the first one I'm going to talk about is acute pharyngitis, or simply pharyngitis. So first one is acute pharyngitis. Pharyngitis, G-I-T-I-S, pharyngitis. So this is or simply pharyngitis. In both these cases, it is most common type of Streptococcus pyogenes kind of infection. So the infection is caused by Streptococcus pyogenes. That's why I've told you that this is a very dangerous guy, because it is caused by Streptococcus pyogenes and it is the most common one. So it is also called streptococcal pharyngitis. Now in this case, it's simply the inflammation of the posterior or oropharynx. We all know that this is the tonsillar area. So it's simply the inflammation of tonsil area, right? So tonsillar area. This is what we call acute pharyngitis. Now it also development of rash, and obviously rash development in neck and also trunk and extremities in response to the release of pyogenic exotoxins occurs in this particular case. And obviously this is very painful situation, because when a person catch this particular kind of symptoms, the eating of food is literally halted in this particular case, right? The second important type of symptoms or disease that they can cause is impetigo. So let me write. Impetigo is the second kind of most common type of symptoms. Now it is usually caused by Staphylococcus aureus, but is also caused by some Streptococcus species also, right? Now this disease begins on any exposed surfaces. Like most commonly they are found to be exposed by surface like legs. So leg surface is much more vulnerable. So let me write, leg surface is much more vulnerable to catch this impetigo symptoms or impetigo diseases, right? And it can cause severe and extensive lesions. Actually, impetigo means simply it's a severe lesion. So let me write simply, it's a lesion that is created, right? Now in this lesion we are having a lot of Streptococcus species staying there, right? And it can be treated with a tropical agent such as mupirocin, right? It can be treated with mupirocin, but still this is a kind of deadly situation, because eventually this lesion will convert itself into what we call a necrosis, or necrosis of tissue. Necrosis of tissue can be resulted in this case. Okay, so that's why it is also dangerous. This impetigo is also dangerous. Now the third kind of diseased condition that can be caused by Streptococcus species, especially Streptococcus pyogenes, is rheumatic fever. It is mostly caused by this hemolytic kind of Streptococcus, but this rheumatic fever means this is a kind of autoimmune disease. Now in this case, this particular disease is obtained due to the cross interaction or cross activity of heart and heart and knee joint antigen, knee joint cell antigen, right? So that these are the major causes. But in these all this kind of rheumatic fever and all these cases, there are always the self reactivity of our immune system. So our immune system recognize itself as a foreign particle, and it is providing the response against itself. So that's a self-destructive kind of disorder. Now it can result in several type of symptoms like rash, like fever, and also arthritis. It is also called the rheumatoid arthritis. It is very common type of arthritis that are found. So in this case, it is going against our immune system, playing with our immune system actually. Okay, so this is the third kind of infection, and the fourth type of symptoms or fourth type of disease that they can cause is streptococcal toxic shock syndrome. I am not writing as simply as toxic shock syndrome because toxic shock syndrome is also caused by Staphylococcus aureus, right? So toxic shock syndrome. Now that's why it is called streptococcal toxic shock syndrome. Now in this case of streptococcal toxic shock syndrome, is the mechanism of toxic shock activity is the same. So it is simply the interaction. It's simply the interaction of a T cell. So it's simply, let me write it, says it's simply the interaction of our host cell, say MHC for example. This is the antigen for example, with the T cell. So let's draw. This is the T cell, right? So say this is the T cell. So simply the interaction. Now in this case, they are producing, the Streptococcus in this case is producing a exotoxin, cell destructive exotoxin. Now this exotoxin here is acting as, it is acting as super antigen, right? It's acting as super antigen. Now as it is acting as super antigen, this exotoxin is replacing the antibody, this peptide sequence or antigen peptide presented by the MHC molecule, right? And as a result of that, it will trigger the activity or response that will call upon many more T cells and different cytokines, which are the chemokine factors or chemical factors, and more T cells. So this ultimately results in propagation of T cells, and finally this chemical mediators like cytokines and leukotrienes and all these things will damage the tissue. So end product of all this thing is tissue damage, right? End is the tissue damage, and finally it can cause disease. Okay, so these are the major four types of clinical conditions that are brought about by Streptococcus species. Welcome back friends. In the previous videos we have talked about the Streptococcus introduction, and obviously we have talked about the diseases that are caused by Streptococcus. Now in this video we'll be seeing the different mode of pathogenesis. So we'll be seeing pathogenesis, right? So pathogenesis of Streptococcus. Now when we're talking about pathogenesis of our Streptococcus, we must talk about virulence of this Streptococcus. We must look at some important factors in all these different bacteria. The pathogenesis is not a single particular part. It is a combinatory effect of different strategies. Now majorly there are two different strategies behind the pathogenesis, and those strategies are, one strategy is to escape immune system, escape host immunity or immune system. This is one type of activity. Another type of activity played by the same individual or organism is the killing or the destruction. So let me write it as a destruction of cells. So destruction of host cell, destruction of host cells. So these are the two different strategies that are taken. And in this case also, these are the two different strategies taken by Streptococcus species. Okay, now the first kind, which is the escaping of host system, is brought about using, this is brought about using, so let me write, take this color, it is brought about using uh, using toxins as well as enzymes, and the destruction of the host cell is also brought about by certain enzymes, and also it is brought about, the spread is brought about using some more enzymes also. Okay, so simply enzymes, so let me write it like that. Okay, now in this case, the toxin that is used in this case is cytotoxin or pyrogenic toxin. So let me write it, it is called the pyrogenic cytotoxin. Now this pyrogenic cytotoxin is also called as, it is acting as actually super antigen, right? And it is going to activate more and more. So if I scroll it a little bit down, it is going to activate more and more T cells, as well as it will produce more and more cytokines, right? So as a result, it will eventually trigger the immune response up in a very little bit. So as a result of that, it will play with our immune system, and it will block the activity of certain immune system, and production of the cytokines and other chemical mediators will harm the tissue in certain regions. Okay, and the second important thing is the production of enzymes. And the enzymes in this case the enzymes I'm talking about are streptolysin enzymes. So let me write streptolysin enzymes. This is called streptolysin O and also streptolysin S. These are the enzymes that are available. Now the streptolysin enzymes will actually degrade cells. It can kill immune cells as well as it can kill normal cells, right? So its lysine means it is going to lyse the cell. Now on the other hand, the destructive feature of the host cell is again brought about by the streptolysin. So as I've told you before, that the streptolysin can degrade cells. So this is one way of doing it. Now other mode of the infection is brought about using the propagation, right? Propagation of bacteria. This is also important during the virulence, right? So if a bacteria, if it need to set the virulence factors, if it need to set the pathogenesis, in those cases it must propagate from one place to another place. And for this propagation, they can adopt three different type of strategies. Actually major two different type of strategies. One type of strategy is the production of an enzyme called streptokinase. So let me write it. The enzyme is called streptokinase. Now the streptokinase enzyme can actually degrade and it can convert the plasminogen into plasmin as a result of the production of plasmin. So let me write it here. So we are having plasminogen, which is the inactive form of the enzyme plasmin. Plasminogen, it will activate plasminogen into plasmin. Now as it activates plasmin, so it will act on it and will produce plasmin. Now as it activates plasmin, this plasmin can break down or degrade some fibrin or other protein network like this. So protein network like fibrin, for example. So fibrin network is there. So this plasmin can eventually degrade this fibrinogen actually, so fibrin clot into smaller fragments. So let us talk about it in a little bit detail like that. So let's say here we are having the fibrin clot. So here we are having fibrin clots coming out like that. Due to the activity of this plasmin, it can degrade it into smaller fragments like that. So fibrin clot is gone now due to the activity of this plasmin. So that's the effect of plasmin, and that is indirectly brought about by the streptokinase enzyme, right? So that's the one way of the propagation. Now we know that this is the fibrin. So if we are talking about fibrin is a protein network that is helping the cells to maintain, the mammalian cells to maintain their structure. Now if it degrades this fibrin clot or fibrin molecules or fibrin network, it will disintegrate the mammalian cells, as well as it can spread intracellularly or extracellularly from one place to another place. So that's the one strategy for the propagation. Now another strategy for the propagation is also the degradation of hyaluronic acid. So this is the second way of the propagation. It is the degradation of hyaluronic acid, right? Now this hyaluronic acid degradation is brought about using the enzyme called hyaluronidase. So hyaluronidase is the enzyme which will degrade hyaluronic acid. So let me draw for you. So if this is the condition, if this is the cell lining, and hyaluronic acid is coming out from this particular side. Let's say this is the site. These are the hyaluronic acid places that is coming out. So hyaluronic acid, as you can see, it is helping them to construct the basal side, right? So it helps them to organize the cells, right? To build the ground substance of the cell so that the cells will make the sheet-like structure, right? So actually the hyaluronic acid. So if this is the base of the cell. So let me draw. If this is the base of the cell. Let's draw. This is the base of the cell. Hyaluronic acid actually holds this base so that the cells are layered perfectly onto the cell. Now in this case, if something cleaves this hyaluronic acid from this region, let me tell you, if something cleaves it from there, as a result the cells will disintegrate. And all the structures that we have talked about, we can see in this case, or in case of mammals, right? So streptococcal infection in mammals will destroy this kind of hyaluronic acid barriers. As a result of that, what it will generate, it will break it down smaller fragments right now, smaller fragments, and as a result of this fragmentized condition of hyaluronic acid is, they can easily spread from one place to another place, right? So these are the strategies that are taken, and there are also two different important strategies. One, to go against this also to go against this immune response, and that is to inactivate the complement system. So they can inactivate the complement system. I forgot to mention this particular part. Let me mention it now. They can inactivate complement system. How? Using or inactivating C5a, right? What is C5a? C5a is an anaphylatoxin. We know that. Now blocking the C5a in this particular case, it will block the signaling of yeah, as it is acting as a signaling molecule during the complement system. Blocking it will result in the blocking the signal of complement in the classical as well as in the alternative pathway. So as a result, less complement activation and less bacterial destruction will occur. And obviously they are having another tendency of production of streptodornases. Now this streptodornase, what is this streptodornase? So it's a kind of new name, streptodornase. Let me write streptodornase. Now the streptodornases are certain kind of enzyme, as it suggests ases. That means it's a kind of enzyme. This is simply a kind of DNase. Simple kind of DNase. Now this DNase that can degrade the viscous DNA in necrotizing tissue. So it can actually degrade the DNA in necrotizing tissue, and simply it will kill those cells during the necrosis. Remember, we have seen that different kind of infection with the help of this Streptococcus will result in the necrosis and tissue damage. In those cases, it will damage or degrade those cells using this streptodornase enzyme, which is a kind of DNase which will destroy those cells by cleaving their DNA apart. So this is another very important mode of activity or mode of pathogenicity shown by Streptococcus, and also a unique one. Okay, so these are the major types of pathogenicity of Streptococcus, and I hope that's helpful. Thank you. Welcome back, friends. In this particular video, we'll be talking about the treatment of streptococcal infections. So we'll be talking about treatment. This is going to be a very short video because the treatment is, again, when we are talking about the treatment of any bacterial infection, the first thing that come into our mind is antibiotics. Simply antibiotics. Now what kind of antibiotics we can use in this case? Usually the Streptococcus species like Streptococcus pyogenes and all these things, they are not still resistant to penicillin, so we can use penicillin G. We can use penicillin G as an antibiotic. Now there are some people which are who are allergic to penicillin G. For those we can use macrolides such as azithromycin or clarithromycin. So we can use azithromycin as well as we can use clarithromycin as the antibiotic treatment. In those cases we can use these two different macrolides. Nowadays doctors prefer this use of penicillin G along with clindamycin because it is going to show us some kind of synergistic effect. So usually nowadays they are using this penicillin G and they are tagging it with clindamycin. And then they use, because these two things show some synergic effect. Synergic effect means when you use both of them, penicillin G as well as clindamycin together, it is going to provide us some very strong positive effect altogether, right? And usually we can treat this Streptococcus pyogenes infections using this combination of penicillin and clindamycin most of the cases. Okay, and obviously penicillin G can be used for the necrotic streptococcus infections too. Okay, so this is, but before treating or before prescribing any kind of antibiotics for treating of the streptococcal infection, we must check for the proper identification of the bacteria from the wound site, right? And then do the rest of the things. And the technique we can use is rapid latex antigen kit, remember. And in this case after knowing all these things, prevention is also most important part, because prevention is always better than treatment and cure. So prevention or preventive measure must be taken. Preventive measure must be taken. So hand washing, and obviously during before the eating of food and all these things, you should be hygienic about your life. Otherwise this kind of infections like streptococcal and staphylococcus infections are mostly transferred by food. So you be careful about food. You regularly hand washing is required. So washing is a kind, and you need to maintain all these things. So that's a little bit about the treatment, and I hope that's helpful. Thank you.